Hicks, and today we're going to talk about amino acid forms. I'm talking about like Zwitterions and anionic and cationic forms of amino acids today. We're going to check out how they are pH dependent, and then we're going to look at an application as electrophoresis as to one way that we can apply this idea in chemistry. First of all, what is a Zwitterion? Well, a Zwitterion is an amino acid that has both a positive and a negative charge on the two ends of it. So I'm looking at this amino acid right here. Here it is in the form that you typically think of it. And you know that on one side of it there is an amine. Amines are basic. On the other side there is an acid, and the carboxylic acid on this side, right? And they are acidic. So because it's acidic, the definition of an acid is that it's a proton donor. So there we see our proton that wants to be donated right there. And so it will donate that into solution. And then a base is something that accepts a proton, right? So it doesn't have to get it from itself. It can get it from anywhere inside the solution, even the water molecules themselves, and it can accept the proton to the amine group. When it does this, the amine group becomes the conjugate acid of itself. And so here it was a base. And now over here that it has accepted a proton, now it becomes a conjugate acid and it has a positive charge on that nitrogen right there. Likewise, over here, the once acidic group now has lost its hydrogen, and so now it is a conjugate base over here on this side. And so the Zwitterion is this ion that has both a positive and a negative side to it. And it is, I want to point this out, it is formed from a conjugate acid and a conjugate base of our amino acid. Now, these are ionic. And because they're ionic, that's going to change their properties a little bit. And one property that it's going to change is going to be its melting point. And because now it is ionic rather than in its molecular form, you know that ionic substances tend to have higher melting points, right? So I'm going to put a little arrow up here to say, hey, the melting points and boiling points of these Zwitterions in their natural environments like this tend to be relatively high. Also, their solubility, because they are, again, ionic. Ionic things love to hang out with water. So their solubility goes up substantially as well. And so being in this form, this Zwitterion form, changes the properties of the amino acid. Now, when we have this Zwitterion, when it's hanging out inside of water itself, water and the environment can be altered slightly, acidic or basic, right? And then that can create a different environment for the Zwitterion. And we're looking here, if it is in an environment that is somewhat basic, then the Zwitterion itself is going to take on, thought I did that once, is going to take on what we call an anionic form, okay? So if it's in a basic solution of some type, so here you can see the OH, it's in a basic solution, then it's going to be in an anionic form. The base here is going to interact with the conjugate acid, yes? And it's going to remove one of these protons, and that'll form our water over here. And now the Zwitterion is left without a charge, without its positive charge, and it only has the negative charge remaining. So that's how it is in an anionic form. Likewise, if we have this in an acidic environment, Then in the acidic environment, the extra hydrogen ions are going to bond with the conjugate base of our carboxylic acid. And here you can see that hydrogen ion has attached itself to the carboxylic acid group here. And now the carboxylic acid group has lost its charge, right? It was negative over here. Now it has no charge. That leaves a positive charge left on our Zwitterion, and so we call that the cationic form of that particular amino acid. Let's take a look at 3 different amino acids real quick. One of the things that you should be able to do is you should be able to draw amino acids in their different forms, either the Zwitterion, the cationic, or the anionic form. So let's practice with a couple of these, huh? Let's take lysine here, and let's turn lysine into its cationic form, or excuse me, its Zwitterion form, okay? So in order to do that, I'm going to have lysine here. It's going to have its N with its 2 hydrogens. I'm going to draw out the 2 hydrogens here just for fun. And then I have my alpha carbon, and then I have my carboxylic acid over here, right? And so with this, then this has an oxygen, and this is an oxygen, but I'm not going to draw the other hydrogen on here because in its Zwitterion form, that other hydrogen is missing, isn't it? Instead, I'm going to put a little negative sign that's over here. This, by the way, just to attach a little more chemistry to it, this little part of it right here is actually a resonance structure with the carbon and these 2 oxygens, right? This double bond here can become delocalized. It can be in either position. So this is actually a very stable form to have this carboxylic acid without its hydrogen, and that's what makes it acidic. That's what makes it so that that proton easily detaches itself because it's becoming a more stable form like this. And over here on this side, the nitrogen will pick up an extra hydrogen on there, and then I'm going to put a little positive on this side to represent that. Okay? There we go. Let's take a look at alanine. Let's draw this one in its cationic form, huh? So this was the Zwitterion form. And, whoops, forgot an R in there. And then with this one, let's draw this one in a cationic form, just for fun. And with the cationic form, I always remember that the cationic form is something that would form inside of acidic conditions, because with the cationic form, I think of the hydrogen ion, acidic conditions, has a positive there, right? And so I attach, in my mind, to remember this, I attach this positive of an acidic condition to that word cation, which means a positive charge. So if I'm making a cationic form of something, I recognize, hey, that's a positive charge. This is something that would be formed in an acidic condition. So if it's formed in the acidic condition, that means the acid part of this, which remember is this nitrogen over here, this still exists, doesn't it? This is where the positive charge is going to be. I'm going to put my little plus here to represent a positive charge. So that's where the positive charge is going to be. Here's my alpha carbon. Oh, I didn't do a very good job with this one, did I? I didn't put in all my CH2s. Sorry about that. I just noticed that. I want to go fill that in. One more here with an NH2 on it, huh? There we go. So on this side here, here's my alpha carbon. I'll put my hydrogen here, and then I'll put my methyl group down here. There we go. And then over here, I would draw my carboxylic acid group. And in this case, I'm not going to leave it like this with its negative, right? Because in its cationic form, that's what's happened. This hydrogen has attached itself to there. So I would draw it with the hydrogen, and I wouldn't put any charge. And then finally, just for fun, let's try this with aspartic acid. Let's do this in its anionic form. And in its anionic form, then I'm going to consider that, again, my reminder system here. I know that OH has a negative charge, so basic conditions here, and I am attaching that word to the anion part of anionic, right? I know which is negative. So since this is negative, that means it's reacting with a base. So what's going to react with the base but the acidic side of the Zwitterion, right? They're going to neutralize each other. So instead of NH3 here, it's only going to be a nitrogen with 2 hydrogens now, because one of these hydrogens is going to go off with the OH and form water. Here's my alpha carbon. And then I'm going to draw the remainder of my side chain here. And that would be another, I'm just going to write COOH if that's okay with you. And then on this side of it, because this does have a negative charge, I'm looking at my Zwitterion here. The negative charge is on the carboxylic end of this. So I'm going to put in my double bonded oxygen, my oxygen here, but no hydrogen. And I'll leave it with a negative charge over here on this side. Okay, so there's a quick couple of examples on how to draw these different forms of an amino acid based upon their acidic or basic or neutral environments. Let's talk about what's known as an isoelectric point for a minute. So an isoelectric point is a point that is where the pH has the concentration so that the Zwitterion, the concentration of the Zwitterion is the predominant ion in there. And it varies, the isoelectric point varies with the side chains themselves. So here's a list over here that I grabbed off the internet. And here is the isoelectric point. We represent it with a pi. This p here actually does correspond to a pH, right? So pH is minus the log of something. So this pi is actually attached to that same idea of pH. If you're taking AP chemistry or something like that, you may refer to, hey, oh, there's pKa's sitting here as well. And so that equilibrium also has a factor in determining what the pi is of an amino acid itself. You can see that it varies, right? Many of them are neutral, aren't they? Are relatively neutral. And for us, neutral would be around 6 or so. I know you think of neutral as being 7, but we tend to kind of think with amino acids that sort of a neutral isoelectric point is around 6. When it starts to get a little bit lower than 6 here, let's kind of take a look at these guys right there. Significantly lower than 6, right? So they have an acidic pi, and then down here I can see these guys down here tend to have kind of a basic pi to them. But most of them are somewhere between like 5-ish and 6-ish, around there. This depends upon their side chains as to which one is part of that, because they can have multiple dissociations of the hydrogens with the side chains if they contain amine groups or if they contain carboxylic acid groups in those side chains. And so that creates the pK2 or pK3 that we have that's associated with those that will then alter the pi of the amino acid itself. So let's see, did I miss anything? If you're in IB chemistry, you'll have a data book that will have this information contained in the data book itself, and be something that you can reference. This isn't anything that you have to memorize. Or if you're in some other course, this would be generally given to you We don't tend to memorize exactly what they are. Maybe you'd be asked to know that when it has a carboxyl group on it, that the pi tends to run acidic, and when it has some type of an amine group on it, it tends to run a little bit basic, depending on the level of your course. Okay, let's take a look at the dependence of these forms, the Zwitterion, the anionic, the cationic form, upon pH, okay? So just quickly, this is an equilibrium. See, there's our double arrows right here. So this is an equilibrium. So you're not going to be just all of one or all of the other. This is dynamic equilibrium, so both of these are present in the solution at the same time. And what we're seeing with this pI and pH dependence here is we're actually seeing a shift of the equilibrium, a Le Chatelier's shift of the equilibrium. So here is a base. So if I make the solution basic, then I'm increasing the concentration of my base here, right? And that's going to shift the reaction over to the right. And that would make it so that the anionic form of this would be more concentrated than, say, the Zwitterion form, or it would be increasing the concentration of the anionic form compared to the Zwitterion form. Likewise, if I make the environment more acidic, I'm going to be pumping that up. I'll shift this reaction to the right, and then that would make it so that the cationic form would now become higher in concentrations more than the Zwitterion, right? It's going to shift the reaction this way. Likewise, if I make it acidic, one more just twist on this here. If I make this acidic, that's going to be lowering the hydroxide ion concentration, right? That would shift this back in this direction, which would cause the anionic form to become less and less, right? And the Zwitterion to become more and more. So really, I have a three-part equilibrium that's taking place here, right? And as I change the pH, the concentrations of the three forms are going to change in respect to whatever changes are being made to its environment. Here's a quick little graphic to try to get an understanding of what's going on just a little bit. So this would be for a neutral amino acid. And so for a neutral amino acid, we can see right here in this form a pH of 6. We said for a neutral amino acid that a pH of around 6 would be that. The pi is the point where the Zwitterion, which is represented in green here, the concentration of the Zwitterion, you can see this is a concentration, right, in moles per cubic decimeter. So the Zwitterion concentration is highest around the pH of 6. This would be for a neutral amino acid that has more of a neutral pi, right? Now, as I increase the hydrogen ion concentration, so my pH is going to be decreasing because I'm going to make this more of an acidic environment. You can see that the Zwitterion form of this decreases in its concentration, and the cationic form of this is going to increase in its concentration, right? What about the anionic form? Well, it's down to like almost 0 here. Is it exactly 0? No, but it's really close to 0. So it's just going to hover down around this line and be a very small part of the equilibrium that's present. This point right here, if you're in AP chemistry, this point right here is a very interesting point. And the reason it's so interesting is because in AP chemistry, we've learned that the pKa, right, the pKa is equal to the pH when the concentration of the weak acid or base and the conjugate are in equal amounts, right? So right here, you can see for this Zwitterion, or for this amino acid, that this is a point right here where the pKa of the substance is equal to the pH. So this just kind of gives you a relationship between the pKa's and what their pHs are in respect to, let's say, the PIs of this, right? And over here we would have a pKb, right? So the pKb is equal to the pH right here, as the anionic form and the Zwitterion form are of equal concentration at that point. So just to bring a little advanced chemistry into that. If you didn't understand it because you haven't taken AP chemistry, you can ignore that for just a little bit. So what do we do with this now? Now that we can change relatively, we're changing the charges of these things, aren't we? By altering the pH of its surroundings. So one thing we can do with this is we can use it in what we call an electrophoresis process. Electrophoresis is what we use to separate things from each other. And as the name implies, we use electricity to cause a separation of the different parts of a mixture of some type. So this is a setup, probably kind of an older looking setup, much simplified looking setup here. Again, I stole this off of the internet. It was the best looking one that I found, very simple. And so we can see that here is some type of a power supply, right? And so we're going to use electricity to cause a separation in these things. And on one side, we're going to have some type of an electrode. And on the other side, we would have some type of an electrode, right? And so on this side, we would call this side over here actually the anode. And that's the positive side of it. Yes, I know that feels weird that the anode is the positive side of our electrophoresis process, but it is. And then on this side of it, the negative side of this is called the cathode side. Many times in biochemistry, though, they'll just talk about it as this is the positive side, this is the negative side. They'll put some type of an electrolyte in here, and an electrolyte is also in here. This is some type of a filter paper, okay? So you can use a paper, you might be able to use a gel in a more advanced setting. And in this paper, we've also soaked it with a solution, a buffer solution that has a specific pH, okay? So let's say maybe we've soaked this in a solution that has a pH of 6. And we would also make our buffer solution, that would be our electrolyte that's sitting in these two containers. They are also at a pH of 6. So this is all one like big electrolyte buffer here that is sitting around at a pH of 6. So being at a pH of 6, that's going to cause some of these ions that are in here to be Zwitterions, right? Some of them will be Zwitterions. And when I create an electrical charge between these two, then the Zwitterion, you remember, has, this is my amino acid, it has one end of it is positive and the other end of it is negative. And so it's really not going to go anywhere. You know, the negative side's going to be pulling on the positive side's going to be pulling on the negative. And so the Zwitterion's pretty much just going to sit there and be stationary. And the two sides of the electrical current is going to be attracting the two charges that are on the amino acid. However, if it was a cationic solution, let's get rid of our negative here. If it was a cationic solution, then this is positively charged, and so the negative side, or this over here, is going to be pulling on that and pulling it towards this side of the buffer solution, or this side of the apparatus. So depending upon what environment we use, the pH of the buffer system, we can change the charges on these particular amino acids and cause them to be attracted to one side or the other in this electrophoresis process. So it's all about using this electric field, a positive side and a negative side, and then having the charges of the different forms of our amino acid, and then using that electric field to attract them to one side or the other. And then they will track through there. Very similar to like if you have done a chromatography lab or something like that, where you had a dye that or something along that line on a piece of filter paper and then put it in water, right, and had it track with the water. It's a very similar operation that takes place here, only here, instead of allowing gravity or allowing the intermolecular forces of water wicking up a piece of paper, here we're going to use an electric charge to cause things to move around inside of our separation process. All right, so here are some things to keep in mind about this electrophoresis process. First of all, we can adjust the pH, and as we adjust the pH, we're going to create charges on these amino acids and on these proteins, right? So if I make it more acidic, I'm going to be turning some of them into their cationic form. If I make it more basic, I'm going to be turning some of them into an anionic form. And then they'll be attracted to different sides of the electrophoresis process. Here's something that's really important to keep in mind. The greater the difference, it's the charge difference between the buffer and the PI. So the greater that difference is, what does that mean? That means more of it will be in either its cationic or its anionic form. So the further you get from the PI in pH, the further that environment is away from the PI, then the more and more of those amino acids are going to be changed into their cationics or anionic forms. And so this is going to allow that amino acid to move even faster because it's going to have more of a charge and it's going to be more attracted to that side of the electrophoresis contraption there. All right, so the greater the difference, the faster it's going to move through the gel or through the paper that we have it set up on. Here's something to note. Not all amino acids are going to have the same PI, right? So this is what creates the separations. Some of them are moving faster or slower. Some of them are moving to the positive. Some of them are moving to the negative. And that's what's going to create the separation that we see in this. Okay, I think I already covered that, so I'm going to move along. So let's consider here a mixture of lysine and alanine and aspartic acid, there we go, and glutamic acid. So let's say we have a mixture and it's all right here. So this is that little glass slide that we were talking about, right? And over here is the cathode side, so it's the negative side, right? And this would be that container that's over here that contains our buffer solution. And then over here is the container that contains the buffer solution as well, right? And on this side is the charge going to be negative. On this side, the charge is going to be positive, which is being supplied by our power supply, right? So I'm just zooming in on that little plate where the separation is going to take place. And we're going to use our imagination for the rest of the contraption here that's taking place, right? The big things to note is where are the charges? Yep, here's the positive charge, there's the negative charge, right? And it doesn't always have to be in the same place, so you need to check your diagram to make sure you know which one it is. All right, so for these here, let's see. I know that aspartic acid here has a PI, I think, of about 2.7. Glutamic acid, I think, is around 3.3. I'm not 100% on that. Let's see, the lysine here is a basic one. I think that's in like the 9s or something like that. And then alanine is our neutral one. and I think that's around the 6. I think I have those right. I can go back and double check them real fast. I just want to make sure. Yeah, alanine is the one that's neutral, and I can see that the aspartic acid and the glutamic acid, right, are around 2.7, 3.2, and then the lysine down here is 9.74, so almost 10, huh? So those are the PIs. So in order to figure out how these are going to separate, notice the first thing that I did was I went and I referenced what their PIs were. So I wanted to figure out what are those PIs for each of the different amino acids so I know which way they're going to go and how fast or how far they're going to go over some unit of time. They will give you some specific pH that's used for that separation. Let's just, in this case, let's just call it 6, huh? And so for a pH of 6, so that's what our buffer solution will be made from. I know at a pH of 6 that alanine, right? Alanine is going to be in its Zwitterion form. So let me just kind of jot this over here. So here I am comparing the PI to the pH of the solution, yes? And so for alanine, I recognize that the pH and the PI are equal to each other, aren't they? And so if the pH and the PI are equal to each other, then that means this is going to be in its Zwitterion form. I'm just going to put a Z there to represent Zwitterion so I can maximize my space. And then next, I'm going to take a look. What about lysine here? So for lysine, I see for lysine that the pH, which is 6, right? So the pH is actually lower than the PI. So listen how I question myself about this. If the pH is lower than the PI, that means the buffer is more acidic than the amino acid, right? So it's more acidic. That means more positive. So therefore, the lysine here is going to be in its cationic form, right? So whenever the pH is more acidic, that's going to make the amino acid go to its cationic form, right? We're matching up the positives with it. So if it's more acidic, so the pH is less than the PI, so if it's more acidic, that means it's going to be positive, and so it's a cationic form of it, yeah? And then the aspartic acid, excuse me, the aspartic acid, that's an S and a P, there we go. I see that the pH here of the buffer solution, which is 6, is greater than the PI. The PI here is a 2.7 for this, right? So because the aspartic acid is 2.7, that means this is in a basic environment. It is more basic than what the PI is, and therefore it's going to put it in its anionic form, right? Likewise, glutamic acid, same thing here. The pH is greater than the PI, which means it's in its anionic form. Now, one thing I want to point out, since I have 2 of them that are both going to be in their anionic form, I want to take a look at the idea that it's about the difference of these 2. So the pH of the aspartic acid here is like very much greater, a much bigger difference between the pH and the PI than the pH and PI of the, that's supposed to be an I there, the pH and the PI of the solution with the glutamic, right? So because this is 2.7, that's further away from 6 than the 3.3 is, right? So there's a larger difference with the aspartic acid than there is for the glutamic acid. All right, So how is this going to play out here with the various amino acids as we run our electrophoresis? Well, the first one that you can figure out real fast is which one stays here, all right? So originally, we put all 4 of them right there. And then we turn the machine on, and it's going to start the separation process. The alanine, where the pH and the pi were equal to each other, it's going to be predominantly in its Zwitterion form. So the Zwitterion has both a positive and a negative charge, so it's just going to sit right here, and it's not going to move. So I'm going to label this, hey, this is the alanine right here, and it's just going to sit right there, and it's not going to move one way or the other. Now, the lysine we saw was in its cationic form, yes? And the cationic form would have a positive charge, and so that would be attracted to the cathode side or the negative side of our electrophoresis apparatus. So that means it's going to move this way, right? And we can just pick some arbitrary distance. I'm just going to say, all right, right here. And this is where the lysine is going to travel to over some period of time, right? Now, the longer I let this run, the further and further and further it will go to the cathode side. Eventually, if I let it run long enough, then all of the lysine will just end up inside of that buffer solution over here on this side. So a person needs to know, hey, I need to turn it off in 5 minutes or something. And a person would judge that based upon how far the amino acids travel and what your voltage is and stuff like that that you're using. So a little trial and error there. For the other ones, the aspartic acid and the glutamic acid, now these are going to be in their anionic form, so they're going to be attracted to the anode side, the positive side, which means they're going to travel this way, right? And I have 2 of them, and I said one of them is going to travel faster in this direction than the other. So the aspartic acid here, with its 2.7, we said it was going to travel further. So let's just put it over here. Again, I'm arbitrarily picking a spot over here, further away, and then I'm going to label this as the ASP, because that's my aspartic acid. And then I also need to put the glutamic acid in here, and I know it's not going to travel quite as far, and so I'll put it here. Again, for all 4 of these, they are all 4 going to be, separated the same amount of time. So the speed, the rate at which it's going, will be represented by its distance, right? So if it goes faster, it's going to end up going further, because the aspartic acid and the glutamic acid are both being separated for the same amount of time. So time would be a constant for these, and speed then could be represented by the distance in which it travels. So this is what my spectra might look like for this or chromatogram might look like for this after I run the electrophoresis process for a little while, showing the different separations of the amino acids based upon what? The pH of the solution and the pi of the individual amino acids at that particular pH. All right, so we've looked at the Zwitterion. We've looked at different other forms of it, like the cationic and the anionic forms, and how they're dependent upon pH. And we looked at the process of electrophoresis, where we can separate these using that unusual property of the Zwitterion and the property of the acidic and basic environments and the side chains of those. So I hope you've enjoyed what you watched. Have fun with your chemistry, and we'll see you next time.